So moving on, super happy to have Simon come and present. As Joe fanboyed a bit earlier, Simon produced one of my favorite movies of plant biology of all time, which is The Caterpillar Eating the Plant. I don't want to tell you in case it's included, ruin the surprise. But he's a professor at Madison, Wisconsin. Like all good biologists like myself, he did his PhD in Edinburgh, but then he moved on to Berkeley and got a position in Pennsylvania. And he does a lot of work on calcium imaging and electrical signaling across the whole plant. And I think one of the coolest things is he sends plants to space. They've been on the space station. So going to move on. So Simon, if you're ready, just share your screen and please start talking.
All right. So we're going to change scales now. And rather than get down to cellular resolution, I think I would kind of classify the kind of imaging that we sort of delve with is putting a whole plant context to give you the questions to ask to get down to cellular context. And so you can ask big questions. When a plant's being eaten, does it feel pain? I think there's a very clear answer to that, which is no. But what we can do is use imaging technology to understand how plants perceive and transmit information about what they're perceiving. And the cool thing is that the imaging technology, GFP technology, has got to the point where for the kind of imaging that I'm going to talk about, it's pretty straightforward. The tools are incredibly well developed. They're incredibly powerful, and they let you begin to sort of focus on the biological questions, which is going to be great. So the biological question that I want to talk a little bit about is this one. All right, so what does that plant know, definitely by know, about the fact it's being eaten? And there's a lot of signals being generated by that caterpillar. There's physical damage, there's chemicals coming out of the saliva. Turns out the plant can even respond to the sound, the vibrations of being eaten. And so I kind of like to know how that works. Like, what does the plant know about what's going on, and how does it sort of deal with that? Because being eaten is kind of an important thing for a plant. So, as was pointed out at the beginning of the whole series, I do a very very small amount of the work that I'm going to show. And so this is the lab, but the two people I should point out at the moment I'm going to sort of concentrate on their work is ex-members of the lab. Masasugu is now a professor in Saitama. He sort of got us into the whole damage world, and I'll show a little bit of his stuff with apparently Alex's favorite movie. And Johnny Johns was a graduate student, is now a postdoc at the University of Missouri, and I'll show some of her stuff. Right. So we know that plants exhibit systemic responses, and I'm just a tiny little bit of a huge field, right? And so we have to keep that in mind. I'm going to concentrate on two stories, two sort of piecing together of some of the molecular machinery which is going on inside the plant. But if you look, there's some recent reviews on plant wounding, and the models are incredibly rich, and there's a lot of fantastic work done by a lot of different people. I'm not going to talk about that. I just want to talk about these two stories, but there is a hugely rich context to place all of this in, which makes the field fantastic to work in. All right, but so what do we know? So an insect starts munching on a leaf, and a lot of people have worked out the immediate response pathway. A lot of that's related to jasmonic acid. We know the molecular pathways. Damage induces a really gorgeous set of responses to defend yourself. Big one of them is the production of jasmonates and jasmonate isoleucine and all of the defense responses that get triggered there. That all happens really quickly. I mean, if you're being eaten, you have to defend yourself almost instantaneously where you're being eaten. Otherwise, you're going to be eaten. But the other thing that we know is that there is some mobile signaling system where non-damaged leaves trigger really kind of similar responses. And so you get this molecular pathway, the cascade of responses. You get a lot of the defenses get induced preemptively. And, if you sort of take a step backwards, it kind of makes sense. If you're being eaten as a plant, the leaf which is being eaten needs to defend itself, but preemptively putting down systemic responses in the rest of the plant is gonna protect you. But again, the timeframes that we have to start thinking about are really, really fast because if it takes you 2 hours to switch on your defenses in a systemic way, you're gonna be eaten. Right? And so this is the fascinating bit that I sort of want to understand is that there is got to be information processing and information transit throughout the plant, and things have to happen at a time frame that is our sort of human time frames. We don't need to necessarily do time lapse imaging in order to capture these events because they have to happen in seconds to minutes. Um, there is a lot of information about what those long distance signals are. Remember, there's a whole field here with really, really clever people doing really insightful experiments. And the one bit that I want to sort of concentrate on is the calcium response because my lab is like fascinated by calcium signaling. It's super, super important and very sort of generic throughout biology. The important thing to realize here is that the calcium ion is not the thing which is moving. The calcium that we're visualizing is the cellular response to these moving signals. So it's a way of reading out the waves of responses going throughout the plant. Calcium itself probably is not well, in fact, we know it's not the mobile messenger. We'll come to that in just a few minutes. So what we want to do is see really rapid responses. The readout of those responses we're going to use is cellular calcium changes. And here, the imaging technology has just opened up the field. So there are a lot of GFP-based calcium sensors. They're all fantastic. The field has matured. The one I'm going to talk about is a family called the GCamps, but all of these sensors are roughly the same. So what GCamp is a circularly permutated GFP with two proteins fused to it. So it's one continuous peptide. The two other bits of it are calmodulin. Calmodulin is a classic calcium binding protein that makes this thing calcium sensitive. And on the other end of the circularly permutated GFP is a thing called the M13 peptide. That is a calmodulin binding peptide from myosin light chain kinase. Right? It was just this interaction was known about when calmodulin binds calcium, the M13 peptide binds to calmodulin. So the way that the sensor works is that when there's no calcium around or very low levels of calcium, calmodulin can't bind the M13 peptide. A great way to think about what the circularly permutated GFP is like is imagine that GFP is a barrel of protein. That barrel is broken apart in the circularly permutated form. The chromophore is exposed to the outside world, to the environment, and its fluorescence is quenched. But when calcium binds to calmodulin. It binds to the M13 peptide that levers the whole thing back together, and the GFP barrel gets reformed, and the fluorophore becomes fluorescent. So lots of different versions of this, but they all basically are that under low ambient calcium levels in a cell, the fluorescence is very, very low. And when calcium levels go up, the fluorescence goes up. And so the readout that we have is we can put this stably transformed into plants. Of course, that's going to be Arabidopsis. And when the Arabidopsis gets brighter, the calcium signals are being generated inside the cells, and you can express it on 35S or ubiquitin, so you can have it ubiquitously expressed. So you can visualize the entire plant. So we're not going to get to cellular resolution, but we're going to see a physiological response played out across the entire plant. And just to give you an idea, and also to put plants back into the context, which I think we all know, but kind of forget about, this is a whole bunch of Arabidopsis expressing this GCamp. This is a movie that actually Matsugu made a long time ago, and it's just one of my favorite movies because it puts everything in context. Because this is real time stroking across a bunch of Arabidopsis plants with a pair of forceps, and they instantaneously light up. Right? So the signaling that we're talking about is our time frame of signaling. I also like it because you remember those experiments where you drop the petri dish on the floor and you pick it back up again and go, I'm going to analyze this plant anyway because it's bound to be fine because plants just sit there. Tons of signaling is generated just literally by taking the lid off a petri dish. Um, for those of you who think that that is not a response to touch, here is a leaf very clearly responding to touch. Again, just stroking the surface of the leaf with a pipette tip. I like it. I kind of cool because it says touch, right? But also, if you look at the response isn't just cells going up and coming down again. There's a wave of response which is emanating from the points of contact. And we'll come back to that because all of this super cellular context and these dynamics are going to be incredibly important for how plants are going to respond. Right. But we don't want to poke things, and we want to know what happens when a insect is chewing on a leaf, because that's the real kind of context that I want to talk about. So, herbivory induces systemic responses. Herbivory induces systemic calcium responses. And so this is an Arabidopsis plant expressing the calcium sensor. You're going to see an insect. The insect is naturally autofluorescent, right? So its green fluorescence isn't calcium, but the green fluorescence on the plant is the calcium levels inside the cells. And so this is sped up a little bit, but like the insect chews on a plant, and both in the directly damaged leaf and in the systemic parts of the plant, there is an emanating wave of calcium response. Remember, the calcium itself is not the thing that's moving. We're monitoring the biological response of cells receiving a mobile signal. That insects are really complicated to understand as they're doing herbivory. There's mechanical damage, there's vibration, there's saliva, there's a lot of signals, but we can trigger the same kind of systemic responses with our lab insect, which turns out to be a pair of scissors. Right? So we can do mechanical damage and we can trigger the same response. And so this is sort of the entry point movie for thinking about what's going on. So this is a Arabidopsis plant. We've numbered the leaves, right? They're numbered by their age, right? And we're going to chop off with a pair of scissors leaf number one, and just pay attention to leaf 6 and 5, which developmentally are very close to each other in their age, right? They're as close as you can get. They're very similar leaves. Chop off leaf 1, and we have our wave of response that moves through the plant. But there is a very distinct patterning to it. And this is where, you can go, wow. But being a little bit more quantitative and a little bit more like developing the patterning and working out the patterning becomes kind of important here, because there's clearly some connectivity within the plant which is playing out here. We can now do some more sort of response molecularly targeted analysis, because we know that leaf 6 gets a calcium signal, but leaf 5 doesn't. And so we can ask, is there a difference between those two leaves? And just I'm going to concentrate on all the imaging side of stuff, but there's a lot of things like qPCR, transcriptomics behind this as well. But just to give you a feeling that the calcium signal does mirror response. If we look at the two leaves, remember, leaf 5 doesn't get a calcium signal, leaf 6 does. And if we look at the induction of defense genes, classic defense genes, we see that leaf 5 doesn't really induce any defense genes, but leaf 6 does. We can do the same thing by measuring the concentration of jasmonate, the defense hormone, and leaf 6 triggers a big peak of the production of jasmonates, whereas leaf 5 doesn't. So that the calcium signaling seems to mirror the patterning of the calcium signal. And so that leads you, that is damaging the leaf. But I think it's really important to remember that this systemic signaling connects all parts of the plant. And so this is one of Johnny John's experiments, damaging the root, right? So we're going to physically damage the root, but we're going to image the whole plant. The root that's going to be damaged is this one down here at the bottom. So we damage the root, and the whole leaf system, the whole rosette lights up, right? So there's communication in all directions throughout the plant. So that obviously leads to sort of getting down to the molecular questions of how is that signal propagated? Um, and there's lots of different ways to answer that. And again, like I say, there's lots of people have done lots of different experiments to work out how the information is moved around. I just want to concentrate on two stories, right? They're almost the same story because one of the things that I'm super interested in is the calcium signal. And if you're interested in calcium signaling, one of the things you're interested in is channels. And so this is a group of channels. They're called the glutamate receptor light channels, highly conserved in plants. Don't try and read anything on that slide, right? That's just looking at the diversity of these GLRs throughout the plants. We're interested in Arabidopsis. I'll highlight the Arabidopsis ones. There are 20 of them. How are you going to study a family of 20 genes? I mean, that is going to be crazy hard to do because there's so many of them. However, remember, this is a field with a lot of really, really good researchers in it. And there's a seminal paper from Ted Farmer's lab where they looked at electrical signaling and they found that two of the glutamate receptor light channels knock out systemic electrical signaling. So, we kind of already have signposts as what we should be interested in. Two channels called GLR 3.3 and 3.6. Uh, if we take the GLR 3.3, 3.6 double knockout and we wound that, nothing much happens. All right. So knocking out these GLR channels blocks the systemic spread. There's some local damage response going on, but the systemic spread has disappeared. Um, the GLRs are ligand-gated channels. They're called the glutamate receptor-like channels because they're homologs of mammalian glutamate receptors. So it doesn't take a huge leap to go, well, what the ligand, if the ligands are likely to be amino acids, maybe it's glutamate. And it turns out the glutamate is a strong candidate for the ligand. And just to give you one example of like, oh, that seems true, the plant on the left is wild type, plant on the right is the double knockout of the GLR channels. We're going to put a drop of glutamate on the tip of the leaf where those arrows are. And in wild type, the glutamate triggers a beautiful response throughout the plant. In the GLR double mutant, glutamate doesn't trigger anything. So it's all consistent with that is seems reasonable. That what we have is damaged cells leak glutamate, and glutamate is a transported signal which is triggering systemic responses. Um, so that's one way that you could say, well, here's one channel, but plants have many, many different channels. And if you're a plant signaling person, the other calcium channel family that you immediately think about are the cyclic nucleotide gated channels, the CNGCs. All right. Um, Again, it's a 20 family member gene family in Arabidopsis. But again, we have some pointers from other researchers in the field. Uh, and there's a awesome paper from the Vadsiri lab that says that as far as herbivore responses are concerned, the isoform that we should be interested in is CNGC19. If you knock it out, you screw up the ability to defend yourself against herbivores. So what Johnny Johns did is looked at molecular responses, qPCR markers, looking for systemic response to wounding, and looked at calcium changes. And it just looked like wild type as far as systemic responses are concerned. It was so annoying. Um, there's a very, very close family member, CNGC20. Johnny then looks at that one, looks like wild type as well. Right? Um, as a graduate student, you are now into the nightmare project. You've got a 20 gene family group of channels, and your two top candidates aren't the right ones. Maybe CNGCs aren't involved. We can knock out a couple of others. There are some pollen express ones, right? Those are probably not going to be important. But what do you do with there's 16 of them left. You'd have to be crazy to look at them all. So Johnny looked at them all. Well, with one caveat that we don't have a good allele for CNGC15. And she surveyed them all for systemic responses, and they all came up negative. except two. Right? And if it wasn't the except two, I would not be giving you talking about this story. So there are two members of the CNGC family, CNC 2 and 4, that appear important. But this is the point where quantification and paying attention becomes incredibly important because I'm going to show you the wild type response again of wounding a root. Right? So we wound a root, and the whole rosette is going to light up, showing you systemic movement of signals. Um, we can do the same thing in CNGC2. I could show you CNGC4, it would look the same, right? And we're going to wound the root, and we get systemic response. And at that point, you might go, oh, the CNGCs are not important. They're not really interesting. But this is where paying attention becomes important. And this is where Johnny goes, I think there's something going on. So let's zoom in on one leaf. And very quickly, we can just look at the response of one leaf. This is wild type. We wound the root and look at the response of the leaf. And we'll get this beautiful spreading wave of calcium going through the vasculature and spreading across the leaf. If we look at CNGC2 mutant, we can do exactly the same experiment. Wound and look, and it kind of looks the same, but you kind of feel it looks a little bit different, right? And we do the same thing for CNGC4. We wound the root, and it kind of looks the same, but it kind of looks a little bit different. Just remember, this is what the wild type response looks like, which is wound and then spread and then waves moving out that disappear. Right? So everything kind of looks a little bit different, but this is where you got to go in and quantify things. And just to we can monitor what do you measure? There's lots of different things you can measure. So one parameter is you can measure how far the wave moves away from the vasculature. Right? So you can say how far does the spread away from the vasculature of each of these different waves, and looking at different orders of branching. So you can look at the midrib, second order. Let's just look at the third order ones, just as an example. All of the vasculature shows the same thing, that when this wave arrives in Colombia, it spreads out from the veins, and in the tertiary veins, about 200, 250 microns. But it peters out in the CNGC mutants. So we can now go the CNGC2 and 4. Well, they don't support they're not involved in that long distance part of it of going through the vasculature. That's what the GLRs appear to do. But they are important for the fine scale spreading throughout the leaf. And that actually ends up impairing their defense responses. So getting down to looking at the dynamics becomes incredibly important now for teasing apart these molecular responses. And just to finish up with the plants are just fantastically responsive to the environment. Systemic calcium waves, right? I've shown you them in response to touch. I've shown you them in response to wounding. Here's another response. This is localized cold treatment. So on the left here, let's go back. On the left here, we have a pipe, a cold source, right, onto this left leaf. But if we locally apply cold treatment, right, we get calcium waves and dynamics moving throughout the plant in response to that. So this is just my ending pitch of just plants are incredibly sensitive to their environment. And, pretty much whatever you do to them, they're going to know what's going on. And that may be playing out throughout the entire plant. So just be kind to your plants when you're doing your experiments. But that seems like a great place to stop and take questions.
Awesome. Thank you so much, Simon. I find it really relaxing watching the calcium wave propagation, actually. And I wonder how many people freaked out when the caterpillar was chomping, suddenly checking their headphones. We should have probably had a disclaimer at the start. I think in the interest of time, we're just going to take two questions. But if there are more questions, if it's all right with Simon, Helen will copy them and email them to you.
Oh, totally. Yeah.
But so the first question is, I hate out reading aloud. This is amazing. Is there a reason why some leaves do not have a defense gene upregulation and some do?
So what the thing which is moving, which comes from measuring the patterns of the vasculature, but also the speed with which the signals are moving. Uh, what we think is going on is that you damage the vasculature, and you have to damage the vasculature to get the systemic component. The xylem literally sucks stuff through it, and the speed with which all of this signaling works, which is crazy, it's a millimeter a second, turns out is the speed of the flow in the xylem. And so the xylem is pulling molecules, and like I say, there's a lot of really clever people who've worked out what those molecules are. Just happens one of them is glutamate. It's not the only one. That gets sucked through, and then it they trigger the downstream things that we measure with the calcium response. The patterning is where the preferential connections, all of those leaves come into the stem, and there are preferentially closer connections between different leaves in the vasculature and how it's all kind of plumbed within the stem. And the patterning comes basically from how closely connected you are. The bigger the damage, the more leaves respond. And you can do the same thing with the higher the concentration of glutamate. Remember when I added that huge concentration of glutamate, the whole plant lit up? But if you go to a very low concentration of glutamate, you get a much lower number of leaves. So there's almost like a volume built into the response system, where if you like scream really loud, the whole plant responds. But if you just kind of go, ow, a very small part of the systemic response gets triggered.
Okay, I'm actually going to cut the rest of the questions off because we're slightly running over time, and Joe needs to tell you about the next session. So thank you very much, Simon, again, and Helen will forward you the questions, and hopefully you can reply to them. And yeah, thank you very much, everyone.
So just incredibly briefly to wrap up, two fantastic talks. I think, we're really trying to help advocate this plant imaging community, but the most important message we want to get across is this is your community. So the more you embrace it, the more you engage with it, the more things we do together, the bigger the community will be, and the better it will be for everybody. So please, and engage as much as possible. And yeah, please feel free to suggest people for talks, nominate yourself for a talk, anything like that would be fantastic. So the next session is going to be on the 16th of July. We're going to have Sven Truckenbrolt, who's going to talk to you about expansion microscopy. And he is one of our he's one of our outside plant speakers. So he doesn't normally work with plants. But one thing that we really want to do with this series is obviously, as well as hear plant imaging and what have you, to see really where the state of the art in the field is, and to hear from these people, and then we can start thinking about how this can be brought in to the plant imaging as well, which is the idea behind this. And the other space we don't know yet. So if you want to talk, get in touch. Otherwise, we're going to start picking people. Absolute worst case scenario. Alex will talk about endocytosis, and I don't think anyone wants that. So yeah, nominate. But on that note, massive thanks to all the speakers. Thank you to the company of biologists for allowing us to do this. We're incredibly grateful. And thank you everyone for coming on, logging in and hearing these two wonderful talks. So have a good day, afternoon, morning, wherever you are, everyone. Bye.
Bye.